2016 we launched it. We're very interested in these seminar series to bring these different institutions together. Our first conversation was, we've got a lot of great science right down here in South Lake Union and in the Seattle area. It's a vibrant community. Let's at least have something that can start to foster this activity. So this time we're actually adding something different at the end. There's going to be a poster session. I'll tell you a little bit more about that later. So today's topic, we've sort of been rotating on different topics, and today's topic is called Emerging Technologies. And so I'd like to introduce our first speaker from the Allen Institute, is Sean Olson. Sean is an assistant investigator in the neural coding group at the Allen Institute for Brain Science, and he joined us in 2013. Prior to joining the Allen Institute, he was a postdoc at UCSD, where he studied cortical mechanisms of visual processing. He received a BS in biochemistry from University of Utah and his PhD in neuroscience from Harvard. Sean and his team are working to decipher neural circuits through a variety of electrophysiological and optical approaches, as you'll hear. And in his talk today, he'll share our work using the newly developed Neuropixels probes, next-generation electrodes that can record the activity of hundreds of neurons in the brain. His talk is titled, "A Brain Observatory for Large-Scale Electrophysiology of Neural Circuits." So welcome, Sean.
Great. Thank you for the opportunity to speak at this symposium today. I'm going to be talking about new technology for large-scale electrical recordings in the brain. The brain is a spiking network. And if we want to understand the neural code that underlies sensation, perception, and cognition, we need to be able to measure the individual spike trains from identified individual neurons. It's been said that the language of the brain is spiking activity. And one of the best ways, highest signal-to-noise ways of measuring spikes is actually through an intracellular recording, which you can see here. This is where you take a patch pipette or a sharp electrode and stick it into the intracellular medium of the cell, and you get a very nice picture of spike deflections, which you can see as 100 millivolt active events, also called action potentials, that propagate along the axon to send information to additional neurons in other regions of the brain. Now, intracellular recordings are extremely difficult to make in vivo, especially during active behavior. So traditional tools for measuring spiking activity are based on extracellular electrodes. And this is historically something of an old technique. So in 1957, David Hubel actually published a science paper describing the fabrication of a new tungsten electrode for isolating quote-unquote single units in the cortex of mammals. And it was this single unit recording here illustrated that allowed Hubel and Wiesel to map out the coding properties in the visual cortex for the first time. And so what you can see here is that in order to record spikes extracellularly, the signal is much smaller, and so the electrode needs to be very close to the cell body where the spike amplitude is largest. So in the intervening decades, additional probes electrodes were fabricated to allow recording from more neurons simultaneously. So we still want the ability to measure spikes reliably from single neurons, but we want to measure populations, okay? And so this is a probe from NeuroNexus, which was widely used in the 2000s, and a decade ago, this is what I use for 50% of my postdoctoral work. So this is a 16-channel electrode, linear array, that is nearly 1 millimeter long, and it can span the depth of the cortex. And typically, a good recording would be a dozen neurons. One would be very happy if you got maybe 20 neurons in a single recording. But this is not sufficient to understand the complex neural dynamics and circuits that underlie behavior, because we know that behavior is generated by very large-scale distributed networks of neurons that are located in different brain regions. For example, in our work at the Allen Institute, we're focusing on the cortico-thalamic visual system to try to understand the neural code that mediates visual perception and behavior by studying these regions here. So the community understands that we need, in order to understand how the brain works, how it computes, how it mediates behavior, we need large-scale recordings from multiple interacting brain areas. And so the Neuropixels probe was developed as a community collaborative effort by the Allen Institute, HHMI, Wellcome Trust, Gatsby, with engineering from IMEC, and many scientific contributions from other institutes. So this really is a step forward in terms of extracellular recordings for several reasons. And it was designed with the rodent brain in mind, although there may be more general applications. So this is what the probe looks like. It's a single linear shank of highly packed electrode sites, each of which can record the electrical activity of neurons in the vicinity of that site. The spacing between sites is much smaller than previously used on probes. And in addition, the number of sites along this shank dramatically is increased. And this is made possible by CMOS technology and the IMEC fabrication. All right. So with a single probe, we can make recordings across multiple regions of the mouse brain. And so here's a schematic showing an example recording in which the probe is inserted through the visual cortex, down through the hippocampus, and into the thalamus at the corresponding visual thalamic region called the LGN. If we zoom in here, we can see that we can record activity across layers in the cortex and also from different subregions within the hippocampus. In total, we can record 384 channels simultaneously from a single probe, and this typically leads to our ability to record from several hundred neurons in a single experiment, which ultimately generates millions of spikes per hour. Okay, so this is a throughput that really is a step forward. In addition, the dense spacing of the contact sites provides a much clearer view of a single spike from a single neuron. And so in addition to the extended number of neurons we can record from, we also get a more precise view of the spikes from individual cells. So here's one example here from what would be called a regular spiking unit. Okay, so this is an extracellular spike waveform that's recorded from one contact on the Neuropixels probe, and it corresponds to this location here. Now, because the sites are so densely packed together, we can measure the action potential or spike on multiple sites around the maximum channel site. And what we see is a spike image, okay? And the interesting thing is that you can see this is shifted to the right. And so if you look at this axis, this is time. So what this indicates is that the trough of the spike is traveling along the probe in the vertical direction over time. And these types of events, traveling waves, have typically not been measurable with extracellular recordings, although there are a couple reports in the literature in which great pain was taken to align the electrode probe precisely with the geometry of neurons. But the thing about the Neuropixels probe is that we didn't go looking for these events, which we now believe are called are back-propagating spikes. We were simply able to observe them reliably due to the enhanced data spatial sampling from the probe. So what I'm showing here is a representation of two different cell types. and their spike waveforms that are associated with those. Actually, the current source density estimate, which is derived from the spike waveform. And again, what you can see in this regular spiking neuron is this traveling wave that moves upward along the probe as the spike is initiated. Whereas for this fast spiking cell, many of which are inhibitory interneurons, they don't show this propagating wave, and they have a much thinner waveform. And so it's these images that can be used to differentiate between cell types in an extracellular recording. And ultimately, we think this can be extended to many different cell types in the brain. For example, in the hippocampus, we see regular spiking waveforms that travel in the opposite direction to the visual cortex. And it's interesting that the pyramidal neurons in the hippocampus that correspond to these spikes are inverted in terms of their morphology. So their apical dendrite here, along which the action potential is propagating, is pointing downward. And again, fast spiking neurons don't show this wave, and cells in the cerebellum, which are known to lack these back-propagating dendritic spikes, also don't show the waves. Okay, so that was a detailed view of a single spike, which will ultimately allow us to think about classifying who we're recording from in extracellular recordings, which historically is a weakness of this type of technology. But in addition, these back-propagating spikes are a phenomenon worthy of study in and of themselves. They've been associated with synaptic plasticity and dendritic integration. Okay, so zooming out from the single neuron level, I want to talk about using a study in which we've used the Neuropixels probe to densely map out the functional architecture of a subcortical region in the thalamus, which was previously not very well explored. And so this is the cortical and thalamic visual system that we're very interested in. We know a lot about the LGN. There's a neighboring structure that is interconnected with the visual cortical areas called LP. It's a homologue of the pulvinar in humans. And so what we did is we made systematic recordings at many different sites within this structure with the probe. And so these are 3 different projections, either a top-down view of this horizontal, coronal section, and you can see these electrode tracks. Each individual black dot is one single recorded cell. And so you can see we can record dozens of cells along an individual shank of the probe. And this is pulled across multiple experiments. But by registering the probe's geometry in the brain to our reference atlas, we can map out how neurons in this structure are encoding visual information. And so we've been able to compile this functional information from all of those cells that we've recorded to generate maps like this, which describe how different regions within the LP nucleus code visual information. So we find several different so-called retinotopic maps where different regions in this structure respond to visual stimuli presented at different locations in the visual field. And in addition, we've found additional functional properties that vary across this structure. Okay, so those were 2 scientific results that we have recently been pursuing based on recordings with a single Neuropixels probe. What we've been working on recently is to develop what we call a brain observatory for doing systematic recordings from different regions of the mouse brain in order to produce an activity map during active visual behavior in the mouse. So the Neuropixels probe is not the brain observatory. The brain observatory is actually a very complex set of individual components that starts with transgenic mice in many cases, involves a surgical step, functional imaging, behavior training, and then we use what we call the Neuropixels platform or rig to do these systematic recordings, and then we collect additional data from the brain of the animal after the experiment in order to register it to our anatomical framework. And so I want to focus in on this step here in terms of a high throughput physiology rig based around the Neuropixels probe. Okay, so this is what our rig looks like. There is a running wheel on which the mouse can freely run. We measure the running speed. We track the eye location of the mouse. We present visual stimuli on a computer monitor here. And then up here are the 6 neuropixel probes, which I'll zoom in on in a minute, which can be targeted to precise locations in the mouse brain simultaneously. All right, so this is a real picture of the rig here. This is a protective shield. Each of the probes is mounted on its own individual manipulator to allow for precise targeting within the brain. And here is a top-down view of 6 Neuropixels probes inserted into the cortex of the mouse brain. So each of these gray shanks here corresponds to 1 Neuropixel probe. And these are targeted to very specific regions in the mouse cortex that we know based on anatomy are interacting and communicating with one another during visual processing. So our ultimate goal is to produce a high-definition activity map within the cortico-thalamic circuitry, spanning multiple thalamic regions and multiple cortical regions, while the mouse performs a behavioral task to map out how information is propagated and transformed along different neural pathways, both between areas and also within a local circuit. So as you can see on the right here, for these 6 neuropixel insertions, they span the cortical depth, allowing us to look at cortical processing within the column in the cortex. And not shown in this diagram, but we can insert the probe deeper into the brain to target the LGN and LP simultaneously, so that within a single recording, potentially on single trials, we would be able to predict the mouse's behavior and which type of visual stimulus it was looking at, simply based on having such a dense recording across the relevant circuitry in the brain that is processing that information. So ultimately, we get very large-scale data sets like this one visualized here. So this is an hour-long recording from a 6-probe experiment from these 6 different visual areas. Ultimately, we recovered 873 individual cells from this experiment, millions of spikes. And ultimately, a major challenge now will be interpreting and analyzing this large-scale data to relate it to the animal's behavior and also the other sensory and cognitive factors that are driving spiking activity within this network. And a big part of because the data is so big and complex, an essential part of our mission is to make these large data sets freely available to the community, because it's ultimately going to take a community effort to solve some of the hard challenges in understanding the neural code in the cortex. All right, so with that, I would like to thank the broad teams, and I'll first start out with people that contributed to primarily to the work that I described today. So Dan Denman and Josh Siegel are leads on the Neuropixels project. The LP and waveform analysis was done by Sam Corbett and Xiaoxin. And our Brain Observatory ops team is over here on the right. Tamina, Greg, and Severine, and Josh is deeply involved in that also. But ultimately, this effort is much broader and involves many more people at the Institute listed here, and even more beyond that, the entire community here. So thank you for listening. I would be happy to take any questions.
get very precise positioning of the probes?
Yes. So we take great pains to, number one, target the probe to a defined region within the brain, okay? And to do that, we have a coordinate system that is anchored both to the head frame and also the recording devices and manipulator system, and also the functional maps that we generate. And okay, my slides went away. I have a backup slide for you. So we have multiple, we try to target a defined region based on the spatial geometry and also the functional organization. And then, absolutely post hoc, after the experiment, we reconstruct the electrode tract in the brain. So the probe is coated with dye, and we use that to identify the precise anatomical location, and then we reconstruct in 3D the location of all 6 probes, okay? And so ultimately, post hoc, there is a processing step that involves mapping the neuron that is recorded on each electrode site to a precise location within our common coordinate frame, this anatomical framework. And as a small side note, I'm really excited about this waveform analysis and this ability to see spike signatures of individual neurons on these probes. And I think it will be particularly useful for identifying neurons recorded from different regions of the brain, especially dense subcortical regions, where they may vary quite a bit in their electrophysiological properties, but their anatomical location is very close to one another. So I think by triangulating various functional features with these spike waveform features, we may even have better anatomical localization. Yeah, in the back.
Grabbing a microphone. So what is the sampling rate for the Neuropixels probe, and how does it compare to more traditional single unit recordings or tetra recordings with some of the commercial or open eFIS systems?
Yeah, so the sampling rate is 32 kilohertz, but we separately collect a spike band and an LFP band. And so that helps with the data compression on chips. We read off 2 different spectral bands, and yeah, so I would say it's comparable. In order to read out so many channels from such a thin shank, that multiplexing was necessary. All right.
All right, thank you, Sean. All right, our next speaker is Elhanan Borenstein. He's from the University of Washington. Elhanan is an associate professor in the Department of Genome Sciences, an adjunct associate professor at the Paul G. Allen School of Computer Science and Engineering, and he's also an external professor for the Santa Fe Institute. So he received his Bachelor of Science in Physics and Computer Science and a PhD in Computer Science from Tel Aviv University, and was a postdoc in Mark Feldman's lab at Stanford University and an Omidyar Fellow at the Santa Fe Institute with David Krakauer before he joined the UW faculty in 2014. His research focuses on modeling complex composition, environment, and function of the human microbiome in healthy and diseased states. His talk is titled Systems Biology and Model-Based Study of the Human Microbiome. So welcome, Elhanan.
So thank you, everybody, and good evening. It is a real pleasure to be here, and I want to start by thanking the organizers for inviting me. And I'm going to change gears completely, and rather than talking about the brain, I'll talk about the gut. And rather than talking about this kind of technology, I'll mostly tell you about computational framework that we are developing in our lab and promoting in our lab in order to really better understand the function, the behavior, and the role of the human microbiome. And so I decided to start this talk with a quote from a relatively old paper about the microbiome, both as a way to give you an historical perspective about microbiome research, but also as really an underlying motivation for almost everything that we do in our lab. And this paper might be a bit older than what you think, because it was written by this guy over here. This is Antony van Leeuwenhoek. He's considered by many to be the father of microbiology, and he used this very simple device, which is really nothing more than a small magnifying glass mounted on a brass plate, to look at different organisms. And back in 1683, he wrote this really beautiful letter, and he started by, though my teeth are kept usually very clean, we have to remember we're talking about 17th century hygiene, so not totally sure what that means. Yet when I view them in a magnifying glass, I find growing between them a little white matter, as thick as a wetted flower. Though I could not perceive any motion, I judged there might probably be living creatures. So we assume that there are microorganisms in his mouse, and he take this flower and he mixed this with rainwater, which we assume were sterile, and then it is said, to my great surprise, perceived that the fourth matter contained very many small living animals which move themselves very strangely, and he goes on to describe them. Right? So this is really the first observation of the human microbiome. And he goes on to describe a really beautiful comparative study where he's comparing the set of bugs that he found in his own mouse to those that are found in kids and women and older people. He wanted to see the effect of xenobiotics, so he looked at the microbiome of a drunk person. And as he said, he was surprised that those animals were not killed by the constant consumption of tobacco and alcohol. And then he ends with this really beautiful statement that the number of these animals in the scarfomentis are so many that I believe they exceed the number of men in a kingdom. This is the first microbiome paper ever. Nowadays, we of course know quite a bit more about the microbiome, and obviously most of what we know about the microbiome has been driven by technology. And maybe the one technology more than any other that contributed to our understanding of the microbiome is what's called metagenomics. So I assume most of you know exactly what metagenomics means, but just to make sure that we're all on the same page, the idea of metagenomics is to study the DNA not from a single isolated and cultured organism, but rather to study DNA from a mixed community of microorganisms. And so the idea is that you take some microbiome sample. If you're looking at your gut microbiome, you basically look at a fecal sample, or you can look at a skin swab, right? And rather than isolate individual species, you extract DNA from this whole community, lyse everything, and extract the DNA. You're getting these mixed fragments of DNA from many, many different organisms. And then you can do usually one of two things. You can focus on a specific marker gene that you believe exists in every organism in one copy, but is different enough to tell us apart different organism. You sequence that. We usually focus on a gene that is called the 16S RNA. We cluster similar sequences together, and by basically counting how many different versions of this gene we find and how abundant they are in the sequence data, we get an estimate of who's there. What is the species composition of the microbiome? Nowadays, we see more and more studies where we are not amplifying a specific gene, but rather sequencing everything that we find. That's what's called shotgun metagenomics. We are getting many, many small bits and pieces of DNA that come from many different organisms, and in most cases, we can't really tell which organism a specific reads come from, we cannot also assemble full genomes, but we map those reads to some functional gene database to get an estimate of what kinds of genes do we have and how many of them do we have. Do we have many genes that are involved in carbohydrate metabolism or in vitamin production, right? And so that tells us the gene composition of the microbiome. And in fact, nowadays, we see more and more studies that beyond just species composition and gene composition are also characterizing many other variables like the transcript in the microbiome, the protein that we find, or the metabolite. Again, all of that at the community level, at the mixed community of all those different organisms. And so by doing that, we're getting a really detailed map or a catalog of the microbiome across many different organizational and functional levels. And by analyzing this kind of data, we can learn quite a lot of things about what is the microbiome looking like and how is it contributing to different diseases. So we've seen numerous studies, probably hundreds of studies just in the last 5 or 10 years, that are looking at the composition of different microbial families in a cohort of individuals with a specific disease, comparing this to the abundances that we find in a set of healthy individuals, and identifying specific species that might be more or less abundant in the disease state. We can do the same looking at the functional data, again trying to see, for example, whether the categories of genes that we find in the metagenome are changing over time or are changing with a specific disease. Nowadays, we see more and more studies that are actually focusing on the relationship between those different omics. How does a set of genes that are encoded by the microbiome correlate with the set of genes that are actually being transcribed? How does the abundance of different species correlate with the concentration of different metabolites, and trying to identify a metabolite that somehow is correlated with a specific species, maybe as a proxy to tell us that this species might be involved in the production of this metabolite. And by using this approach, we now know quite a bit about the microbiome. So first of all, we obviously know, and probably many of you heard that we're actually mostly microbes. If we estimate the number of cells in the human microbiome, the number of microbial cells outnumbers the number of human cells 10 to 1. We know that the microbiome is extremely complex and diverse. If you look at your hand, you're probably looking down about 150 different species. If you look into your gut, you might find close to 1,000 different microbial species. We know that the microbiome is extremely variable. So the species that you have on your left hand are actually very different than the species that you have on your right hand. The species that you have in your gut are very different than the species of the person sitting next to you. And the species that you have in your gut today might be very different than the species that you'll find in a week's time, specifically if you change your diet, for example. And what's probably is most exciting, that when we are doing those kind of analysis to identify the shift that we find in specific disease, we find that the microbiome seems to have a role in many, many different diseases, and this is really just a partial list, ranging from the obvious suspect like obesity and diabetes, all the way to autism and cancer and cardiovascular diseases. But at this point, I want to argue that with this great advancement in microbiome research in the last 5 or 10 years, really the most fundamental questions are still very much unresolved, right? We don't really understand why do we see different set of species in different individuals with a specific disease or across time. We don't really understand what are the forces that are determining the composition of the microbiome, and maybe more importantly, how does those shifts change the activity of the microbiome and ultimately the physiology of the host? Does it matter if you have more of this species or less of that species? And most interesting of all, if we think about a translational application, we don't really know how do we design an intervention that is beneficial, right? How do we move the microbiome to a more healthy microbiome? How do we engineer a new microbiome that is capable, for example, to address problem of malnutrition? And what I want to further argue that the part of the reason for this gap has to do with the way that we study microbiome data. And I'll go back to this von Leeuwenhoek study, and I'll try to contrast this with what we do now. So in terms of device, obviously we're using a slightly more complex and much more expensive machinery. In terms of the scale of the data, von Leeuwenhoek was able to characterize 5 or maybe 10 species, and we now at every metagenomic assay are characterizing thousands of species and often millions of different gene families. But when we think about the analysis, we're really doing exactly the same thing that von Leeuwenhoek did. We are comparing lists. We are comparing catalogs of species, of gene, of metabolite between different cohorts. And I always like to think about it as if we're trying to understand what is the difference between an iPhone and a Galaxy phone, and the only thing we do is put both of them in a blender, cataloging the different component and saying that's what makes an iPhone so great, if you're an iPhone person, is that it has 3 more screws and maybe 5 more transistors. And we're completely ignoring the mechanism, the wiring diagram, how different components interact with one another. And so to try to address that, what my lab is trying to do for the past 8 or 9 years is really to introduce more methods that are inspired by system biology, by modeling, with the rationale that if we'll start to focus more on interaction between different components, on mechanisms, on how different types of data should relate to one another, we'll be able to learn much more about the microbiome and maybe understand a bit more how is the microbiome contributing to health. And so we're doing everything from trying to build a metabolic model of the microbiome that would mechanistically explain how the microbiome digests your food and how does the food change your microbiome. We're using models to analyze data, and we're applying this model to a very large set of different data sets. And so for today's talk, I'll mostly focus on this line of work in our lab when you're using a model-based approach to analyze data and maybe more importantly, to integrate different omics. And so as a quick motivation, I'm going to take you back to this slide that I showed you before and know that all those studies that are analyzing multi-omic data are generally, if they are integrating the data at all, if they do that by statistical association. And when we saw that, we realized that not only we have the traditional problem with association study of association does not mean necessarily causation, but also I want to argue that association does not promote understanding, right? We don't really know what is happening. And maybe more importantly, it's an extremely inefficient way to study the data because if you're just looking for association between different omics, you're basically ignoring everything that you know about the biology, the ecology, and the genomics of different systems that actually links different omics to one another. And so what my lab is trying to do is take a completely different approach as follows. So we think about different processes that links different omics to one another. For example, we think about what we know about metabolic control and about enzymatic activity, and how what we know about these processes should link or should affect the set of metabolites that you find in a microbiome sample to the set of protein and the set of genes that you find. We think about what we know from ecology and species interaction, how the presence of one bug in the community could affect the behavior of another. And today I'll show you mostly studies where we focus on maybe the two most common types of omic in microbiome data, those on the top, and show you what we do in order to build a model that describes this kind of interaction and then use this model as part of the analysis. And so again, I'm going to focus on studies that have species composition and gene composition, and I'll try to build a model that link the two in order to analyze them simultaneously. And so again, just to iterate the link between species composition and gene composition, assume you have some microbiome sample. It is composed of a mixed community of microorganisms. Of course, what makes them different is not the different colors, but the fact that each bug is coding a different set of genes that are marked here as letters. And as I mentioned before, using different analysis approach, you can either profile the taxonomy composition or the species composition of the microbiome and basically create those pie charts of who's there, right? Or you can use shotgun metagenomics and basically completely ignore which species are there and just characterize the abundance and presence of different gene families, right? And again, you can create those pie charts that will tell you what kind of gene categories you find in your sample. But it's important to note that these two profiles are not independent. And in fact, the thing that links them is simply genomics. Put differently, if we know what is the set of genes that are encoded by each bug in the community, we can create a very simple mathematical model that will tell you that the abundance of each gene family at the metagenome is really nothing more than a linear combination of the copy number, how many copies of this gene are present in each bacterial genome, weighted by the abundance of this genome in the community. Right? It's a very, very simple model. And so we have here a mathematical model in this case that is linking species composition to gene composition through genomic data. Now, this is an extremely simple model, and I assume and hope that every researcher that is doing microbiome and metagenomic research somewhere in the back of their mind are thinking about this kind of relationship. But what surprised us at the lab was how rarely this model is actually being used when analyzing the data. And what we wanted to show is that by looking at those relationships and using this model, we can actually infer a bit more when we have this kind of multi-omic species and gene information. And so I'll show you a few examples. And the first one is actually the last one that we developed. This just came out, I think, a month ago. This is an interactive browser that is called Burrito, right? And what Burrito will show you after you input the various information, it will show you the abundances of different species or different clades of microbial species. You can zoom in and out across your samples. It will show you the abundances of different gene families. Again, you can go up and down in the level of description, but it will also allow you to see all the links between every species and every gene. And so whenever you select a species here, it will actually highlight in the functional profile which part of the metagenome was attributed to this specific species and how much of the gene family is attributed on average and many, many other statistics. And so rather than exploring these two data sets independently, you can look at the relationship between them. Another thing you can do, which was done by Sharon, a grad student in the lab a few years ago, is to try to find cases where this model actually doesn't work, right? When what we expect to find in the metagenomes based on what we know about the genome content of each species and the abundance of each species doesn't fit, right? And there are multiple reasons why we can have this breakdown of the model, but maybe the most important one is strain level variation. In other words, my assumption of what is the gene content of a specific species is different between individuals. And so Sharon constructed this beautiful pipeline that is analyzing metagenomic data and trying to identify those mismatches. And by doing that, she was able to characterize how much strain variation there is in the gut microbiome across a large cohort. And the main result that I want to note is that there is a lot of that, right? In other words, the strain of Erectale that I have in my gut might be very different from the strain that you have in your gut, not only in terms of small SNP, but in terms of which gene it's actually encoded. In some species, almost 25% of the genes in a genome of a specific family is actually completely different between different individuals, right? And so that again was done simply by looking at when this model does not work. Just to name a few other kind of a few other studies that are using this model. Several years ago, a postdoc in the lab showed that in cases where we actually don't have genomic information, in other words, your microbiome has species that you don't have reference genome for, you can use this link between species, genome, and metagenomes to deconvolve the metagenome and basically figure out which gene must come from which species in order for these equations to hold up. Another postdoc used this model to renormalize microbiome data and in fact showed that by using this normalization approach, you are seeing differences in different microbiome with a much stronger signal than before. We also show that if you account for this model, that can actually explain one of the canonical and puzzling results in microbiome research, where you're seeing individuals that have very different species composition, but very similar gene compositions. And finally, just a couple of months ago, another student in the lab used this model to characterize and to predict the robustness of microbiome. How sensitive is a microbiome to a perturbation in the composition of species that you have? The last study I want to talk about in a few more details, again, something that came out, I think, about a year ago. And it is trying to take this idea of modeling the link between species and genome and metagenome back into this domain of comparative analysis. And in fact, it came from a discussion with a close collaborator that was studying a specific disease. He had a set of controls and a set of cases, as I described before. He used taxonomic profiling. He defined the composition of species in each individuals. And he was able to identify specific species that are differentially abundant between the cases and the control. He was also able to do functional profiling, again, characterizing the gene composition, and again, found specific gene families or gene categories that are more abundant in the cases. And what he wants to know is, can you explain the link between these two? In other words, I don't care what is contributing this gene to this metagenome. What I want to know is, why do I have more of this gene or more of this specific pathway in a specific disease? And can you identify the species that is responsible potentially as an intervention target? And I have to admit that when he asked us this question, we came back and said, no worries, we'll get back to you in a week, because we had this great model that linked species to gene to metagenome. It turned out to be a bit more complex than that, because once you're looking at shifts, a lot of the assumptions that we made in the model did not hold. And so after 2 years, we came back and we said we actually solved it, right? And so what we developed is another framework by Ohad Manor. It is called FishTaco, for a reason I'm not allowed to disclose. But basically what FishTaco is doing is accepting information about the taxonomic profile, the species composition across a large cohort of cases and control, information about the gene composition across this cohort. If you have reference genome, we use those. If not, we're actually inferring them using the deconvolution approach I mentioned before. We're applying several statistical methods and several methods from machine learning. We're applying a method from game theory that is called the Shapley value analysis, which is really a beautiful way to characterize contribution of species when you have multi-species communities. And by the end of the process, we're basically addressing this exact quantity, which is how much does each species contribute not to the abundance of a gene, but to the shift in the gene abundance between different samples. And so just to give you a sense of how does a FishTaco run look like, this is analysis that we done on a cohort with type 2 diabetes. If you compare the metagenome, the gene composition of individual with type 2 diabetes to a healthy individual, you find genes that are associated with 3 different sugar transport to be more abundant in the type 2 diabetes patients, right? And if that's the only analysis that you've done, you can't really tell much more than that. If you have information about the species profile, you may be able to identify species that are differentially abundant. But what is the link between the two, you really do not know without FishTaco. If you apply FishTaco, you will have this. It will basically take this enrichment and will break it down to the different microbial player. It will tell you the reason that you find more gene of this specific sugar transport is about 10% attributed to change in the abundance of Escherichia and 80% to the abundance of Firmicutes. In fact, there are also some species that are trying to kind of rescue this imbalance, but there's just not enough of them in the community. What got us really excited was that when we tried to see how does that translate to other pathways or to other sugar transports, we see a completely different contributor profile. which means that although we see 3 different sugar transport, all of them are overabundant in type 2 diabetes patients, the underlying species that is causing that is bringing those genes with them, are very different. And again, in terms of translational application, it means that if I want for some reason to rescue this imbalance but not touch the other ones, there is a very clear intervention target. Right? If you change the abundance of Escherichia, you might be able to rescue this specific imbalance. And we analyzed many, many different data sets, and this idea of very specific species contributor profile repeated again and again. This is just one more example. We looked at a database again of type 2 diabetes, but also one of inflammatory bowel disease, compare them to normal samples from healthy individuals. And you see that both of them have an enrichment of methane metabolism pathway. So the same kind of functional enrichment, but in different diseases. If you apply FishTaco, again, you see that the underlying play are completely different. So even though the functional imbalance is the same, if you want to target that, you might need to address a very different set of species. So I'll just say that FishTaco is available online. You can download that. You can run this on your data. We have a web application that actually generates these plots, and you'll start seeing more and more paper having this FishTaco plot, which always amuses me. I also know that all the software that developed in our lab, and I didn't talk about most of them, are always available online, and you can use them. I just want to maybe note very quickly that this approach of linking genomes or using a model to link different omics is not limited to genomes and metagenome and species. This is another framework that is called Mimosa, where we build a model to try to link the composition of species in the community all the way to the concentration of metabolite that we find in a stool sample. And what we showed is by looking at a simple model that is looking at genomic data, metabolic data, reaction data, and building a very simple metabolic model, we're in fact able to predict which individuals should have higher abundances of metabolites, at least certain metabolites, simply by looking at the ecology of the microbiome. Right? And so we use this model, again, to link metabolic information all the way to species information. And so I thought I'll end here. Whenever I talk about modeling to any kind of audience, I'm always assuming there is a bunch of you in the crowd that are saying, but these models are wrong, they're incorrect, you're missing a lot of things, there are a lot of processes that are not included in your model, and this is all correct. But instead of trying to convince you this is still useful, I'm going to quote from 2 of my heroes about modeling. The first one is Arthur Rosenbluth, that said that the best model for a cat is another cat, preferably the same cat. And I want to emphasize that that was written in irony. You should never use your own cat to model your own cat, because if the model system is as complex as the original system, you can't really learn anything. But maybe what's more important is a quote by George Box that said that all models are wrong, but some are useful. And so I hope that I was able to convince you that even though the models that we're using are incomplete, have errors in them, have missing information, I think they're still extremely useful when you're trying to analyze the data. And so with that, I'll end. I'll thank all the students and postdocs that work on that. I think I mentioned most of them by name, the funding, and everybody here for listening. And I'll be happy to answer any questions.
Thank you. Fascinating. I have a question. Most of this data is stationary. It's a static view at that particular moment, right? So we still have that main question: what came first, chicken and egg? You know, question: is the change in microflora the cause of disease, or is disease changing the microflora? And actually, both could be correct, and probably is correct at both processes. And then, of course, you have the interaction between the host and the microflora.
Right.
So you know, that's probably one question that is really important. And then, if some of this stuff that you are showing is correct in terms of cause of disease, we may find out that at some point that at least in some % of people, for example, type 2 diabetes is an infectious disease. So you know, it's this particular shift in flora and in metabolome in people who are in a process of developing diabetes, probably the most interesting. So maybe people who are who don't have disease yet, but they're at the
Right.
risk would be maybe an interesting.
So I'll start by saying that everything you said is completely correct, right? This problem of causation versus an artifact or a side effect is in many cases not clear, and I actually agree that in most cases it's probably bidirectional. There are cases where we actually know that it's causative, and this is mostly done not through prospective studies as you described, which are being done, but they're still rare simply because you need an extremely large cohort. But there are a lot of studies that are using transplantation into a germ-free animal and showing a causative effect. And obesity is a good example because obesity, you can transfer obesity by transferring an obese microbiome. But also if you change your diet and you change the amount of calories and you change even the distribution of different macronutrients, you completely change your microbiome. In fact, what we are trying to do now in this part of the lab that I did not describe is trying really to see if we can model all that mechanistically. Why, if you're having more protein in your diet, your microbiome will change in this direction, if you're having more carbs in another direction. I would say that in general, longitudinal studies are unfortunately missing in the microbiome literature, and they are being generated as we speak, but the problem is, as a computational biologist, I know exactly what kind of data I want. I don't think I'm going to see it soon because I want a very dense temporal resolution, I want a large cohort, and I want a very long time period. And usually one of these is missing for obvious reasons. And so we do have some longitudinal data, we do have some transplantation experiment, and so I think the causation is starting to come out from, at least for specific diseases. In some cases, even for diseases that you may not suspect, but we're seeing that by transferring a microbiome, we see some effect. But there's a long way to go, definitely. Yeah.
I take your point about your last slide. I was wondering if you've been able to quantify the effect of missing or miscalculated species and genes.
Yes, the problem is that there are a lot of things that are missing. Starting from the sequencing that is probably missing the very rare species. And one thing, for example, that we don't know is do rare species have an impact or not? Right? If you have a species at 0.0001%, could it still have a crucial role in the gut microbiome? And the reason we don't know that is because we don't understand the mechanics of the microbiome. There is tremendous amount of information that is missing in the model. Maybe before that, the biggest problem that we have now is missing gene annotations. Whenever you try to annotate metagenomic data, somewhere between 30% to 50% of the reads are either not mapping to anything or they're mapping to gene that we don't know their function. Right? One of my students wrote a paper on that at some point and called this the gray matter because everybody's talking about the dark matter, those species that we never sequence. I'm more concerned about the species that we sequence, but we don't understand what their genes are doing. And so in some cases, we are trying to do kind of a robustness analysis, starting with what we do know and trying to scale back and see how much that affects a different aspect of the microbiome. A lot of the methods that we have are quantifying the confidence of our prediction or how much of the signal is not accounted for by what we have. I'm not sure we can always do that extremely accurately, but at least we're trying. Okay, thank you.
Thank you. Okay, our final speaker for today is Rob Pierce from the Fred Hutch Cancer Research Center. So he walked over a couple blocks, which is great. He's a scientific director of the Immunopathology Corps and a member of the Immunology program in the Clinical Research Division. He received his BA in philosophy from Yale, then earned his MD at Brown University, and then he completed a residency in anatomic pathology, followed by a postdoc fellowship in environmental pathology and toxicology right here at the University of Washington. So prior to joining the Hutch, he was a fellow alumni from Merck and focused on the development of tissue-based biomarkers for Merck's anti-PD-1 therapeutic antibody. His lab is focused on mechanisms of tumor-induced immune tolerance by which tumors can escape anti-tumor immune responses, including natural and therapy-induced responses. And his talk is entitled Parsing the Tumor Microenvironment, Multiplex Immunohistochemistry, and Beyond. Welcome, Rob.
It's a pleasure to be here today. I've walked by this building many times and not come in, and I've often thought maybe they have answers to the questions the problems that I have. So I'm hoping to get through a bunch of pretty images pretty quickly and then just lay out some of the problems that the field still needs to solve and invite you to help solve those. So, and I have just completed an interesting full traverse back to academics from having been an academic at the University of Rochester, went into industry for 10 years at the DNAC site in Palo Alto. If you know immunology, you're probably familiar with the contributions of that site. And now am back at the Hutch. My role at the Hutch is pretty unique in that I was told not to worry about getting my own grants at first. That my role really is to catalyze collaborations with other investigators and to bring these technologies to bear so that we can better understand tumor immunology and create improved therapies. So this is a multiplex immunohistochemistry and beyond. The beyond part I really want to, as I said, enlist your help with. I want to talk briefly about where we were for decades, where we are now, and where we are heading. Now, if I were to die today, this would probably be my legacy. It's the 22C3 and me story. My group at Palo Alto did develop this companion diagnostic while at Merck. And this is the classic problem of immunohistochemistry that we've been trying to overcome for years, and we're still stuck in this model to some degree. This is a single color DAB stain showing PD-L1 in lung cancer, and it was very useful to help develop and get an approval for pembrolizumab in lung cancer. And so although not without its uses, I've been trying to move beyond 1+, 2+, 3+ pathology data for my entire career. And I think we're actually getting to the point where we can do this in a very reproducible way. So this is where I think we are, and this goes back to a paper that we published in 2014 where we had developed this 22C3 assay to measure PD-L1. And just as an aside, one of the tremendous issues in all of immunohistochemistry is trying to know if your antibody is really staining something on target. You can show that it's staining something not what you expect it to be, but to really be convinced that your antibody is behaving is really one of the great struggles of immunohistochemistry. The field was fraught with disinformation, frankly, because most of the antibodies that had been made up until then, save Li-Ping Shen's PD-L1 antibody that wasn't available to us, cross-reacted to a hemopoietic antigen that was present everywhere in the tonsil. And it wasn't until we actually took apart the tonsil and did flow cytometry with multiple anti-PD-L1 antibodies and knew exactly in the tonsil where the staining was. And it was on the deep crypt epithelium and the follicular macrophages. Until we had that Rosetta Stone, we didn't know that that there was all this disinformation about PD-L1 and where it was in cancers. But with this antibody in hand, the 22C3 antibody, we began staining some tumors, and this was probably the biggest aha of my career, where just looking at some melanoma samples where we knew that this PD-1 stain was associated with CD8 T cells and the PD-L1 stain was associated with myeloid cells in the tumor. And we looked at this dual stain using this fluorescent tyramide approach that I'll describe in a minute. We saw that there was this tight interdigitation of PD-1 positive CD8s and PD-L1 positive myeloid cells. And in one fell swoop, what came to you is this notion of adaptive immune resistance, that CD8 T cells were going into the tumor, seeing their antigen, upregulating PD-L1 because of the production of interferon gamma. And this is really basic immunology 101. Upregulating PD-L1 and putting the brakes on an active anti-tumor CD8 response. So patients who had this pattern, this tight proximity of PD-1 and PD-L1, we thought would respond to pembrolizumab. And patients that looked like this, unfortunately, are the majority of patients in most solid tumors where they do not have an active CD8 T cell infiltrate, don't respond. Now, this was the contribution we made to this part of the study, was this stain. Paul Tomei, a dermatologist who was the first author on this paper, he didn't have access early on to this antibody. And what he did was instead look at CD8s and their relationship to the tumor especially at the invasive front. And so very laboriously, he hand drew a line here at the margin between the tumor and the stroma and counted CD8s in this invasive front, this region, and found that there was a cut point above which patients responded to pembrolizumab, but below which they did not. So although it seems pretty basic at this point in time, this, I think, really underscored to the community that microanatomic location matters in tumors, and furthermore, that the proximity of one cell type to another matters. Well, you know, we probably always were cognizant of this as scientists, but this actually mattered very much in terms of opening up billion-dollar markets in immuno-oncology. And although I wouldn't say this was the cause, but it certainly correlates with a vast investment in trying to understand the tumor microenvironment and the development of technologies to do so. So this is where we're at now. Rather than, you know, 2 color stains, we routinely perform 6 antibodies, and plus DAPI as a nuclear counterstain, so 7 color stains on formalin fixed material, and it certainly is pretty. But I think beyond, let me just describe how this is done. It's basically just employing a standard HRP conjugated antibody, or in this case a polymer, as a detection method that lays down substrate. But rather than that brown DAB, we can use fluorescent pyramides of different flavors and then get multiple colors on one tissue. Now what we do is come in with a primary antibody, a secondary antibody that's actually a polymer conjugated with HRP. We add these colored fluorescent pyramides. The HRP activates the pyramides. You get a free radical that then binds to closely adjacent reactive macromolecules on the tissue. And then we come in and basically do another antigen retrieval step. It removes the antibody and HRP complexes, and then we do this over again. So a second time, another color, and we get a deposition of both red and green here. And we can do this up to, with our current technology in the lab, we can do this up to 6 antibodies. And then you get this deposition of many, many different colors there. Now, to resolve these requires spectral deconvolution, which you're all familiar with from flow cytometry, I'm sure. So this is the equipment we need. To do these assays by hand, I think, is against the Geneva Convention. It takes days to do. This is our Leica Bond automated stainer. Even on this robot, it takes about 20 hours for us to do a 6-plex assay. And here's a picture of the Vectra. And then downstream of that, it's how do you collect, and you have to do spectral deconvolution to isolate each color individually, and then you have a lot of data that you have to sort through. And as I'll show you, we use a very user-friendly image analysis program called Halo from Indica Labs. So it's not just a pretty picture anymore. It really is embedding tremendous amounts of data, but to get to that data, you need to be able to first, in our current technologies, decide what is a cell and define where that cell begins and ends. And this is a process called segmentation. And once we have segmented those cells, identified all those cells, we can basically specify them across the entire tissue slice, where here is like cell 1, and then all of the different markers associated with those, plus the intensities of those markers. And anyone who does a lot of flow cytometry knows that this is really a list mode-like data set. And by virtue of that, we can employ some of our old friends as flow cytometric data analysis to look at some of these. And this can be quite useful, especially when we try to assess thresholds of what is really a positive and what is really a negative. Now you can notice here, if you're adept, that if you're looking at CD4 by CD8, we see here CD4s over here, CD8s over here, and then this funny population of double positives, which we don't really think exist at that kind of frequency. And this is one of the problems that I'd like some of you in the audience to help us solve. This sort of, when you're looking at tissue, having these cells smushed together, when you're trying to segment them and say what belongs, what pixel belongs to one cell, what pixel belongs to another, when they're really jammed up closely, you can get occasional false calls. And this is just another example of CD20 and CD3. We really don't think that CD3 and CD20 are found on the same cell ever. This looks for all the world like it's a CD20 positive cell, but when you overlay CD3, you see that actually this is a CD3 positive cell. So though good and reproducible and taking the subjectivity of a pathologist's eye out of it, you do have some inherent problems with this kind of image analysis system. So other things we can do that are really valuable, once we have this list of all these different cells and what qualities they have, is you can ask, okay, how about a double positive, CD3 and BLIMP1 or some other marker that you have, and then that will be mapped by this software into on this dot, say here, and then you can do some interesting spatial analysis of these unique phenotypes. So what's more, this is what I call a Scooby plot because it's so psychedelic. And it's really useful, and this makes easy what was hard even in 2013 when Paul was actually measuring the CD8 densities at that invasive front. Now this software can do it automatically very, very quickly. You can define how big these bands you want them to be. It will, this looks like that one artist who used to put things around islands. It will then score all of the cells of interest in each of these zones. And here's an example of showing that CD8 densities differ as you move through these zones. Other things it can do is calculate nearest neighbors. It will look at the distance from a CD8 to its nearest tumor cell. It will look at the average neighbor distance, the number of unique neighbors, and all of this can be very useful in understanding what's going on in the tumor microenvironment. This is a current list that's always changing of the antibodies that we've validated for multiplex IHC. I should say that the experimental histopathology group is open for business, and if you have any panels that you'd like to do, we're always looking to expand this list. So I just want to talk about our workflow that we're using to understand the tumor microenvironment, and then give two vignettes. So what we often will do is get an HD of a tumor, to assess the adequacy of the sample. We'll do nanostring transcriptional analysis because we find that that's really the most robust in the face of formalin fixation. And then we use that to kind of sculpt which multiplex panels we'll use to further query the tumor microenvironment. And then we move that into Halo for the kind of analyses that I just showed you. A new workflow that I'm very excited about involves the complementary approaches of single-cell RNA-seq and multiplex IHC. And I'll show you more about those in a minute. So I just want to talk about some work that we're doing together with Antony Rangvo, who's a couple doors down from me over at the Hutch, and we're looking at the role of macrophages in driving metastases in cancer. Now, he's brought with him from Yale a very interesting humanized mouse model called the Mr. G, as opposed to the Ister G, the difference being MCSF. These are cytokines that are human cytokines knocked into the mouse in the mouse loci, so that they have the appropriate tissue distribution of those cytokines. And you can see here that the Mr. G has a lot more human CD45 engrafted than the Ister G, which is a decent mouse in and of itself. And most telling is here in this orange box here, which are the myeloid cells in the Mr. G. So there's a full complement of macrophages in these mouse that are not present in other humanized mice. And here you can see the macrophage staining, human macrophages staining in the liver and the lung in the Mr. G, but not in the Ister G. So if you implant a melanoma that's metastatic, the M275 line into these mice, here you can see the melanoma is labeled in green with this cocktail of antibodies. Another cocktail of antibodies in red labels the human macrophages. The incredible thing about the difference in these mice, when you have a complement of human macrophages that are matched to the cell line, which is human, you see a wildly metastatic phenotype, liver, spleen, there's Mets in the lung as well. In the Ister G, this metastatic phenotype is significantly suppressed. So what's going on there? Well, in order to examine this, Antony did a single-cell RNA-seq on these mice and basically found a unique cluster, which we call cluster 4 here, which is in the Mr. G mice, not in the Ister G mice, and is in the tumor-associated it's in the tumor, and they have transcripts showing that they're macrophage lineage, and this appears to be the unique population of cells that confers this metastatic phenotype. So what we did to try to understand that further is look at some of the genes that were pulled out of the single-cell RNA-seq to look at what genes here might we find candidate IHC antibodies that specify this population. And we're in the process of validating these antibodies as IHC reagents and working up a multiplex IHC assay so we can go back to the Mr. G versus Ister G, test that we're actually identifying those particular macrophages, and then analyze some human tumor samples to see, are they there, do they really exist in human tumors, and do they confer an aggressive phenotype? This is where we've gotten so far. This is an example of a 2-plex where we are staining for Marco and CD163, and that looks pretty good. And we're overlaying now other antibodies that correspond to those important differential genes in that population. So another vignette, again, to show this workflow of and how complementary single-cell RNA-seq can be with multiplex immunohistochemistry. This is a fabulous example coming from Kelly Paulson and Ode Shapui of translational immuno-oncology. And just this is a little complex, so it'll take a bit to describe. This is a patient who had multiple previous lines of therapies, had Merkel cell carcinoma, which is an aggressive cutaneous malignancy. Most Merkel cells are driven by Merkel cell polyoma virus, as was this patient. And this is basically showing this patient's tumor burden. He received some TIL that were specific for an HLA-B restricted polyoma virus antigen. So these were an adoptive T-cell therapy that saw this particular antigen presented in this particular HLA molecule. And he also got CTLA-4 and PD-1 therapeutic monoclonal antibodies. And you can see here that after this therapy, he enjoyed a very deep partial response to about 90% for a very long period of time, but suddenly began to recur. And these indicate biopsies taken through this course. And what we can see over here are some MIHC analyses of before he received the combo immunotherapy, again with that adoptive TIL that saw the antigen, that where you see this incredibly cold tumor. There's not a CD8 infiltrate really in the tumor at all. And here, as the patient's responding, a biopsy was obtained, and you see the CD8 infiltrate into the tumor, and it looks moth-eaten, like they are killing off the tumor. And then later, when the patient is progressing again, this patient has reverted to this low TIL or this immunoquiescent phenotype. So what's the mechanism of this? Well, Kelly was able to do some very nice analyses. With HALO, we identified that, in fact, the TILs, in the pretreatment, there's few TILs both in the tumor and in the stroma. In the post-treatment, the TILs go up. And then in the recurring lesion, so on progression, again, this is showing that the TILs now have left the tumor again. And you can see that quantified here. So again, she used single-cell RNA-seq. And this is an overlay of a biopsy from the pre-treatment and then the tumor at relapse. And you see that these populations of host immune cells overlap almost completely looking at the single-cell RNA-seq. So this Tisney plot is really showing the relationship across the entire transcriptome as measured by this assay. But you see that the tumor changes radically from pre-treatment to relapse. And if you look in here, some of the genes that correspond to this change happen to be HLA-B. And if you remember, the target of this adoptive cell therapy was a Merkel cell epitope presented by HLA-B. And you can see amongst all these different changes within the tumor phenotype, what you see is the selective loss of HLA-B in this patient, whereas you don't see a loss of HLA-A. Kelly went on to show that actually if you remove those tumors, you could recover HLA-B with treating with 5-azacitidine or with interferon gamma, showing that it really is an epigenetic modification leading to HLA-B suppression. So those are just two vignettes that I think really underscore what we can do with this current technology. And here, what's beyond this? And this is where I really want to enlist your input. Let me just move to this list of questions here because I'm running out of time. So some problems and limitations of our current MIHC, I mentioned in passing. You know, the fluorescent tyramide approach leads to covalent binding of tyramide substrates. It requires unmixing, and that means you have limited optical bandwidth. So it constrains the number of analytes. We could do 6 antibodies now. PerkinElmer has come out with a new platform that can do up to 9. But the computational unmixing the spectral unmixing is pretty computationally heavy. And that means on our system, you have to have a region of interest that you defined for that to then get spectrally unmixed. And that puts a potential selection bias into your assay. What's more is if you're looking at unmixed images to pull out those regions of interest, and that can be sometimes a challenge. The spectral scanning is slow, and the use of repeated heat-induced antigen retrieval or epitope retrieval to strip the antibody-antigen complexes, that can affect the antigenicity. So it's a bit of a very empirical process to try to get these assays dialed in and validated, and that takes quite a bit of time. There's a long duration. I mentioned how long it takes to do these sequential staining steps, 20 hours on the robot bond, so that actually constrains your workflow bandwidth. The equipment that we need is pretty expensive. And then the technical challenges I mentioned with antibody validation. What's more, this approach is limited to 2D rather than 3D, and I think we need to go there to really understand the relationships within the tumor microenvironment, and even 4D to look at those relationships over time. As I mentioned, there's imperfect segmentation, and although this quantitative image analysis with Halo is wonderful compared to previous iterations of this kind of approach, it's still laborious, and it's limited to testing user-derived hypotheses. And I would like to move to a system where potentially machine learning can generate hypotheses that I don't see within the tissue. And of course, translational oncology requires access to high-quality tissue, which it really is what will move the field forward. So those are the problems, and we can discuss potential solutions. So this is just to thank Antony and his team, which we've collaborated on the Mr. G mouse model. Kelly and Ode and their team on the Merkel cell front, and then the rest of people working in my lab trying to develop new technologies, and the experimental histopathology group that really runs this multiplex platform. So with that, I will take questions. Okay, no questions. Huh. Excuse me?
Here.
Do you often have a problem with cells overlying each other? Is that what's causing the segmentation problem?
I don't think so, because we're working out of 4 or 5 micron thick sections, so it's very planar at that level. All right.
I think we're
Great, thank you.
finishing up here. Let's give a round of applause for all of our speakers again. And as I mentioned before, this time we're trying something a little different. We've got a poster session that's up with refreshments in the lobby, and then I guess the posters are just up the stairs there. So please, everybody, join us there. Thanks.